So, we will switch over to this topic, tumor immunology. It's a very important topic. Look at some of the historical facts. Immune system basically protects us from all the diseases, including cancer. But this concept was not kind of clear for quite some time. As you can see, historically, what is called Ehrlich hypothesis. Ehrlich and co-workers somewhere in 1909, they were one of the first to hypothesize that the immune system has the potential to recognize and destroy tumor cells. Then again, Thomas and McFarlane Burnett, this is also important, in the late 1950s, kind of accepted this and reiterated this idea that the so called pre-neoplastic cells, you know what are pre-neoplastic cells? Though after all, the cancer cells are actually originating from the normal cells. They are not alien cells. They are our own cells which are getting converted from their normal status into transformed status. So, such cells which have the potential to be transformed and which are in the process of getting transformed into cancer cells are called pre-neoplastic cells. So, whenever some changes take place, they said McFarlane and Burnett and Thomas, they said that these cells can be identified by the immune system before they are visible clinically, before any clinical symptoms appear. They can be killed by the immune cells. So, this is known as theory of immune surveillance, because these cells keep a watch on those cells which may get transformed into cancer cells. So, that is the theory. It is not quite accepted as usual. People do not accept once you say some theory, once I hypothesize something, because they have their own doubts. Now, as I told you, the genes are many genes are associated with cancer, they are not alien genes, and so their products are also normal proteins. The main idea behind immunological surveillance is that it can distinguish between self and non-self. So, on the one hand, we are saying the cancer cells are actually hailing from our normal cells. On the other hand, we are saying these cells can be recognized by the immune system. So, this was not acceptable, because immune system means that will distinguish self from non-self, and we are saying the cancer cells have originated from normal cells. So, how the immune system will recognize? That was the question. That is why this hypothesis, although it was correct, but it was not accepted initially. So, that is the question, how then the immune system would recognize the neoplastic cells. So, people did not accept the so called Burnett theory. There is another reason, that the tumor incidence in immunocompetent and nude mice was more or less same, but now we know the reason why this happened. But before that, I will ask you a simple question. You understand what is meant by immunocompetent and nude mice? Immunocompetent means the mice have the immune system, so they are competent to deal with. But nude mice, what is nude mice? Immunocompromised system. Why? Because we are removing the T cells, mostly thymus is being removed. So, thymus removed, but now we know that it is not completely eliminating the immune system. Nude mice is not as an absolute immune deficient system, no. That is why this difference, but that was not known for some time. So, people were arguing why immunocompetent and nude mice, why nude mice and immunocompetent mice are different in tumor incidence. So, that is the question. That is because the nude mice is not an absolutely immunodeficient system. So, that is the reason. Now, we know that immune system can indeed recognize pre-neoplastic cells, and several evidences have been provided by experiments in animal models and in humans based on several observations. I will tell you some of the observations. Basically, the immune system continuously monitors and destroys aberrant cells. However, some cells can escape from this monitoring or evade, or they might have developed alternate strategies to overcome this surveillance. Basically, the immune system tries to kill the cells which are deviant, but the cells develop certain mechanisms to overcome this immune surveillance. So, there is a fight between these two. Whoever wins, the direction may change. Either the cells may not become cancer cells or they will become cancer cells, depending upon the capacity of those cells to fight with the immune system. So, that is what we have to understand in an overall. If you want to have an overall idea, it is something like a almost like a war between immunocytes and cancer cells, and recent studies have identified several mechanisms or strategies from both sides. So, this is what is a tumor immunology that we are going to study about. So, what are the important questions? So, we need to answer 3 questions to gain a good understanding of tumor immunology. What are those questions? Number 1, how the immune system protects us from getting cancer, or in other words, what are the molecular mechanisms of surveillance and protection? What are the mechanisms? See, we know that we are simply saying immune system protects us, but that is not what we are going to study. We have to study what are the actual molecular mechanisms known that are involved in this immune surveillance. So, that is the first question. Second, why in some cells the immune system fails to protect us, or in other words, how do cancer cells escape these protective mechanisms? What are the mechanisms employed by the cancer cells to overcome or to evade immune system? So, that is the second question. Third question, this is a typical question, what is the use of all this? Will it help that if we know these mechanisms, what is the use? The ultimate use is, can we make use of this in developing anti-tumor therapies? Do you understand the logic? So, there are only, if you try to read tumor immunology from textbook or any other source that you can lay your hands on, try to get answers for these 3 questions, then you are okay, because this is fundamentally what we are attempting in tumor immunology. So, giving this background, we will go to the other things. See, as I repeatedly told you, I always assume that you have an idea about immunology and cell biology, molecular biology and all that, because I cannot be teaching those subjects. So, I am assuming that you know what are NK cells. They are natural killer cells, very good. T cells means you know that. TC means cytotoxic T cells, then TH means helper T cells. Reg, have you heard of? Regulatory T cells, very good. Macrophages and dendritic cells. So, these you must also know what is innate immunity, acquired immunity, cellular immunity, humoral immunity. These terminologies you should know before we proceed. Although I am not a big immunologist, but you must have some basic idea before we dwell into, because when I say some molecular mechanism, unless you know these terms, you won't be able to appreciate what is going on. So, that is why I just put these sentences here. Now, we will look at how what are the molecular mechanisms by which we are protected, that is evidences for immune protection. The first evidence is, we now know that there are tumor antigens. That means tumor cells have some antigenic proteins, right, some antigens. First observation is mice immunized with chemically induced tumors were protected against subsequent re-challenge with the same tumor transplanted into them. That is, you give some chemical and induce tumor, and subsequently if you do that, because they are immunocompetent mice, they have some protection. You see that they were called transplantation rejection antigens. Those antigens second time when you are trying to give, they are getting rejected. So, transplantation rejection antigens. Similarly, experimentally induced tumors with different chemicals, carcinogens, viruses, UV radiation and spontaneous tumors show the same outcome, same thing. This kind of if you challenge them again with the same agent, they show some protection. Then T cell deficient mice, T cell ablation or T cell deficient mice removed this protection. So, I told you that there is some protection when you challenge the mice with chemicals, UV rays or virus or whatever methods that you are inducing cancer, there is some protection. But if you have removed T cells, then this protection is not seen. That means what? Immune system is protecting, because you are proving that by removing the T cells. Transfer of T cells from an immunized mouse could protect a naive mouse from tumor challenge. I hope you understand this. That is, if you have already injected something into the mice and if you have taken the T cells from that, because they have been immunized already, and now we introduce that into a normal mouse, then you are able to see this protection. So, that is what is shown here. So, mice were more susceptible to carcinogen induced or spontaneous primary tumor formation under the following conditions that now we know. I told you nude mice is not a perfect system. So, what is a perfect system? Mice lacking interferon gamma receptor or the STAT1 transcription factor required for interferon receptor signaling, this is a good system. These are some latest systems apart from the nude mice. Then RAG2 minus minus, that is RAG2 null mice. I hope you know what is meant by null mice. Completely lacking T cells, B cells and natural killer T cells. So, this is a very good system, natural killer NK cells, you know. So, if you make mice with this kind of a system, then you can see the difference between immunocompetent and immunodeficient. That is, in other words, nude mice is not a completely perfect model of immunodeficiency, but RAG2 null mice is a very good system. So, in that system you will see the difference between the immune protection. So, ultimately proving that immune system does protect us. See, this is an experiment to prove that immune system will protect us. How will you demonstrate that experimentally? You do experiments in the presence and absence of the immune system. That is what it is. Then you see whether there is protection. If there is no immune system, there is no protection. If there is immune system, there is protection. So, that is what is all these experiments, whatever we are saying, these are all just explaining this. So, this is the most important evidence for us to believe that immune system really protects us. In human tumors also, you see tumor specific antibodies. That means we have the immune system, because you have a particular tumor and you see some antibodies against that tumor specific antigens. So, that means you have the immune system protecting you. The system tries to protect. It is another matter that it may fail, but it still tries to protect. That is what it is. Long back also people have found what are known as tumor infiltrating lymphocytes. Pathologists have observed that whenever you see the tumors, they saw blood vessels around that tumor, especially the tumor infiltrating lymphocytes. By pathologists long back also they have seen that. So, that means why this is happening? That is because the immune system tries to protect. Not only that, such patients were better protected. So, they had improved survival as a higher tumor infiltrating lymphocytes if you have, and you also have the cancer, then your survival is better. So, that means it is clear that lymphocyte tumor lymphocytes, infiltrating lymphocytes protect you, that is the thing. Lymphocyte proliferation in draining lymph nodes were observed in melanoma and breast cancer. So, these are all evidences that immune system protects us. There is another interesting observation. Some tumors like some melanoma and lymphoma, they spontaneously regress. How you have not treated them, but they regress. How that can happen? Only through immune system. Our common sense tells us that it must be something is protecting them. What could protect? From our knowledge we know that it is the immune system. So, this is another evidence. Another thing, regression of metastasis also after removal of the primary tumor, pulmonary metastasis from renal carcinoma. So, these are all some evidences. Higher incidence of cancer after immunosuppression, that happens. You see AIDS patients, they are immunodeficient. So, it is said that they have more propensity to get cancer. So, if you are immunodeficient, you are very likely to get cancer. So, that is the idea. So, that also again tells us that immune system is protecting us. Not only in that, if you are given immunosuppressive drugs, there are many disorders, diseases where people are given immunosuppressive drugs. So, they also are susceptible to get cancer. So, that is again another evidence. Aging is a natural thing. It is said that your chances of getting cancer increases with increase in your age. As you become older, you are more susceptible to get cancer. That is because your immune system also gets weakened when you are getting older. So, this is another evidence that immune system protects us. So, I am just giving you some of the mechanisms, some of the things that we may be able to understand how the immune system protects us. So, there is another thing, immunosuppression leads to increased development of virally derived tumors like Kaposi sarcoma, non-Hodgkin's lymphoma, HPV related cancers and so on. So, virally induced cancers, because you see once the immune system is not there, the virus can easily invade you, because the viral, whenever you have viral infection, it is the immune system that protects you. And if you are already under immunosuppressive drugs, naturally the virus finds it easier to attack you. So, that is simple as that. Organ transplant again increases malignant melanoma risk. This is also another evidence. NK or the so called gamma T cells, T cell loss also correlates with the increased tumor pathogenicity. So, these are all some of the evidences people have given. How the immune system distinguishes the cancer cells from normal cells. We have already told this question. Cancer cells basically arise from normal cells. Hence, the many of the proteins expressed by cancer cells are also normal proteins and are not expected to trigger any immune response. So, then how to say that the immune system recognizes? That is because a few proteins are structurally novel, not exposed to the immune system during development and are thus antigenic. This is one mechanism. then the major mechanism is mutation. For example, mutated RAS oncoproteins or mutant p53 can be recognized by the immune system as foreign, and cells that express these proteins can be targeted for destruction by the immune system. In other words, when the proteins are structurally changed, which happens during the carcinogenesis, the immune system can recognize them. So, that is the idea. Although we are talking about they are all coming from normal cells and all that, but the proteins from those normal cells have been structurally altered, and this structural alteration makes the immune system to recognize them. So, that is the idea. The first point I told you, I am going to give an example, that is cancer, what did I say, not exposed to the immune system during development. We will give some examples for that. I will tell you as we go along. Tumor cells also accumulate numerous mutations during the course of their rapid proliferation. So, this eventually can give rise to several structurally altered proteins that can be targeted by the immune system. That is one thing. Another mechanism is defects in DNA mismatch repair. I have not told you what is DNA mismatch repair, but we will talk about it later. But just understand, this will create what is called microsatellite instability. I hope you know what is microsatellite instability, which results in high rate of mutations. So, cancers that exhibit this microsatellite instability have a higher degree of T cell infiltration and lower rates of metastasis. So, that also again tells you that the immune system can recognize. Then there are what are known as tumor specific antigens. They are present only in tumors, but not in normal tissues. For example, this mutant RAS, mutant p53, and the so called I told you about this protein, where you have come across this PCR-able Philadelphia chromosome I talked about, right, in CML, AML, all those kind of things. So, what are those antigens? Some are the tumor specific antigens and so on. Some are differentiation antigens, such as the melanocyte differentiation antigens, p53, mutated p53, that happens due to. So, the antigens can be from differentiation or by mutation or by overexpression. So, the tumor specific antigens can be from different sources. One, they can be arising from differentiation or by mutation or by simple overexpression, or even they can be alien proteins like viral proteins. That is an exception. Normally, you are getting it from your cells, but sometimes if you have viral infection, viral proteins may get into your system, because viruses may have some oncogenic proteins, I mean genes, viral antigens, such as this human papilloma virus, many viruses. Overexpressed cellular antigens, we have already studied this, so called HER2/neu, human EGF receptor 2, HER2/neu antigen. So, these evidences clearly disprove the notion that tumor antigens are simply overexpressed normal proteins and therefore cannot elicit immune response. You remember I told you they are all just proteins, our own proteins. So, how the immune system can recognize? That is not a correct idea, because many of them are structurally altered, many of them have undergone mutation, or sometimes they are overexpressed, sometimes they are hailing from viruses. So, immune system can recognize, that is the idea. Now, we have talked about tumor specific antigens. What is the difference? The tumor specific antigens are those which are present only in the tumors but not in normal cells, whereas the tumor associated antigens are not limited to malignant tissues but are also present in normal cells. So, there are two examples: tumor specific antigens, tumor associated antigens. Now, we will see some examples. Normal proteins that have not elicited immune tolerance due to various reasons. For example, proteins that were exposed only during embryogenesis. We have one example that is called carcinoembryonic antigens, CEA. This is what is called oncofetal antigen, that is, you see that in the fetus, that was not exposed to the immune system because it was in the fetus. So, that is one reason. Proteins that are sequestered in areas inaccessible to the immune system are not presented to the immune cells due to the lack of what are MHC class 1 molecules? Major histocompatibility complex, you know that. So, MHC class 1 molecules. This is because you see one of the examples given is testis. Testis is usually in the scrotal sac outside the system, outside the body. So, during development it may not see the immune system, some of these proteins. So, the ovaries also sometimes they are in separate bag like structures. So, such molecules are possible. The one other example is cancer testis antigen that are expressed in germ cells of the testis and ovary, but silent in normal somatic cells. There are some examples: MAGE, M-A-G-E, and N-Y-E-S-O, NISO, you can say NISO1. So, these are some examples. I want you to remember some of these names as they are very important in the immune context, immunology. Molecular mechanisms of immune protection against developing tumors. We will talk about those things, how our system fights with the immune system a little bit later. We will go one by one. So, both innate and acquired immunity, they work together to eradicate tumors. Several in vitro experiments have been done. However, of course, the challenge is to prove them in vivo. One of the things is natural killer cells. Natural killer cells play an important role in killing the cancer cells. So, this is one mechanism. The FC receptors, I hope you know what is FC receptors, CD16 and all that, present in NK cells, recognize the antibody bound infected cells and attack them. NK cells also can kill hematopoietic cancer cell lines and virally infected cells in vitro. They have shown that the NK cells are very, very efficient. There is what is called NKG2D receptors which are present on NK cells. This is another mechanism. They may be activated by their ligands present in cancer cells. So, once they are activated, they can kill the cancer cells. The natural NK cells can kill the cancer cells basically. This is one mechanism. Similarly, interleukin 2 activated NK cells are also good killers. So, NK cells have several mechanisms. NK cell deficiency is linked with increased incidence of Epstein-Barr virus. EBV means Epstein-Barr virus associated lymphomas. What are the other killers? Macrophages. They can recognize some of the tumor surface antigens. So, they can engulf them and they can kill them. This is another mechanism. Activation of macrophage by interferon gamma produced by the tumor specific T cells. What are the other killers? Actually, the killers are listed in the last sentence here. In the macrophages, TNF, tumor necrosis factor, lysosomal enzymes, reactive oxygen species, nitric oxide. These are the actual molecular killers. These are the killers. Reactive oxygen species can kill, nitric oxide can be a good killer, TNF, tumor necrosis factor, lysosomal enzymes. These are the actual killers in many systems. T lymphocytes also can play an important role. In virally induced tumors, you know the antigen presenting systems and all. The oncogenic peptides are presented by the MHC1 system, then CD8 positive T cells can kill them. Once they are presented by these cells, CD8 positive T cells can kill them. That is another mechanism. Tumor infiltrating lymphocytes from solid tumors, they contain what are CTLs, cytotoxic T lymphocytes. So, this is another mechanism. Similarly, helper T cells specific for tumor antigens may secrete cytokines such as TNF alpha and interferon gamma that can increase tumor cell class 1 MHC expression and sensitive to, they can sensitize the cells to CTLs basically. CTLs are cytotoxic lymphocytes. So, by secreting these factors, they can sensitize the cancer cells to be killed by CTLs, cytotoxic T lymphocytes. So, these are several mechanisms. Finally, last but not the least, antibodies that we have discussed. The antibodies may kill tumor cells by activating complement system or by so called antibody dependent cell mediated cytotoxicity. What is happening there is the Fc receptor bearing macrophages or NK cells can mediate the killing. I hope I have explained this earlier, but this is also this granzyme pathway is there, perforin, cytotoxic T cells via secreting the perforin can activate what are known as granzyme B and granzyme A. So, apoptosis can be activated through caspase 10 activation and then caspase 3 activation, that happens through granzyme B. If you have granzyme A, then there is a lot of proteins what are called, I am not going into the details, what are called set complex that can enhance DNA cleavage, again through apoptosis you can kill. Basically you are, this mechanism is aimed at killing the cancer cells through apoptosis, and that can be initiated by two pathways, granzyme B which mainly activates caspase 3, granzyme A which mainly activates DNA cleavage, both are needed for apoptosis. So, this is a very important mechanism. Yeah, this I have already told that animal models, particularly the mouse models, have shown that the immune responses impose a selective pressure on the cells in such a way that some cells die while some clones of reduced immunogenicity survive and get selected for better survival. What this means is the cells undergo some sort of editing. We call that as editing or so called immuno editing. What it means is, if a tumor is induced by a chemical carcinogen in immunocompetent mice, then the derived cancer cells are said to be edited. I hope you understand this. That is, the cells, if you have mice or other animals which have the immune system, they have the capability to edit the cells, because they have the immune system. If you do not have the immune system and then you have put some cancer cells into them, if you have injected cancer cells, then they cannot be edited, because they do not have the immune system. So, that they are said to be unedited. So, that is what it means. If the tumor is induced by a chemical carcinogen in immunodeficient mice, then the derived cancer cells are said to be unedited. Simple to understand. If you inject cancer cells in mice which have the immune system, immunocompetent mice, the derived cancer cells, suppose you are taking back the cancer cells, you are injecting in a particular area and then you are doing a surgery and taking back those cancer, you have transplanted, basically you have transplanted cancer cells in a particular area, say in breast, and then you are taking back those cells, the derived cells, then they are said to be edited, because they have seen the immune system. What is happening is, the immune system has somewhat kind of three phases of immunoediting. That is, if you understand this, it makes it simpler. Three phases of immunoediting. What do you call by immunoediting? Immunoediting has three phases. One, the elimination phase, that is, innate and adaptive immune systems work together to detect the presence of a developing tumor cell and destroy it completely before it becomes clinically visible to you. That is elimination. That is, immune system can do this. It can completely eliminate, but it also fails, right. So, what happens that rare tumor cell variants may survive, some resistance may take place and they can survive this immune system. They are said to be in equilibrium. Elimination phase, they have crossed the elimination phase, they have not gone into the elimination phase, rather they survived. Such cells enter the equilibrium phase, in which the adaptive immune system basically prevents tumor cell outgrowth and also shapes their immunogenicity of the tumor cells. So, they are kept alive somewhat in a dormant way, they are kept alive, they are not destroyed completely, they have escaped, but not escaped completely, they are somewhere in between, they are said to be equilibrium. The third one, they completely escaped, they have developed some mechanisms and they have escaped. So, you have three types. One, the cells are succumbing to immune system, they get eliminated, they get killed. Second, they survive the immune system, get edited, but they are somewhat in between, they have not completely escaped the immune system. Third, they have completely escaped the system and continue to grow. So, in the escape phase, tumor cells that have acquired the ability to circumvent immune recognition and or destruction methods emerge as progressively growing visible tumors. So, they will start aggressively growing, because they do not care about the immune system, they have defeated the immune system. So, in other words, there are certain cells which are completely eliminated, there are cells which have completely overcome the immune system, there are some cells which are in between. That in between cells are said to be at equilibrium, that is what it is. They have recognized this. We will kind of stop here.